reactive oxygen, which is really not a very nice thing for the plant to do, and they're completely stripped of everything. Their cell walls, all their nutrients, the plant takes the whole lot. And the only thing that's left is the DNA. Now this is where it gets interesting. The DNA travels along up into the main part of the root itself, stimulates the formation of root hairs, which is what you can see in this photograph here. These little strands coming out are root hairs. The DNA travels along the root hair and is basically spat out back into the soil by the plant. And then when it's out there, it reforms a cell wall and becomes what it was before. That's the bit that really gets me. So a bit of naked DNA reforms a cell wall and then goes and loads up again with whatever it is that the plant wants. So it's a cycle. And you can read up about this in the literature, or I can send stuff to Julia and she can send it out to you. So plants nurture microbes in the root zone. They enter the root tip via the soft meristem cells, which are just on those newly growing roots. They're exposed to reactive oxygen. That breaks down their cell walls, releases their nutrients. Those internalised microbes stimulate the formation of root hairs. They exit the plant by the root hairs. They start the cycle again. So that's the rhizophagy cycle. Now, I have to leave time for questions. So I'm just going to talk for another, just 5 minutes. 5 minutes, because I do want to, I did promise you questions. So the endosphere, this is the last. So we've done the phyllosphere, the rhizosphere. The endosphere is the bit. Is that correct? Oh. I thought I started at 9:30 and I only could talk till 10:30. Oh, anyway, I'm not going to argue with you because if I've got more time, that's fantastic. Okay. All right, I can slow down now. Right, so the endosphere. So the endosphere is the internal workings of the plant. And to tell you the truth, I never actually thought about all the microbes that were in plants until recently. So I used to teach in the Department of Agronomy and Soil Science, and then I taught first year science in the Department of Botany, and we did lots of taxonomy, and we looked at photosynthesis, and we looked at heaps of things under the microscope and talked about all these processes that happen in plants. We never ever talked about the endosphere. Just like I suppose in human health, we never talked about the human gut, right? The gut microbiome. We just forgot about the microbes. Well, the microbes that live inside plants are probably the most important ones. So inside plants we have two different things, well, lots of things going on, but the most recent research has been about the core microbiome, in other words, the microbes that actually belong to that plant, just like you have a core microbiome, and every animal has a core microbiome, every plant has a core microbiome. That's what was in the seed. And when the seed germinated, those microbes came to life and helped that plant establish and start talking to the microbes in the soil as well. They set up communication channels. So you can imagine what happens if you actually put insecticide and fungicide on that seed. I mean, the core microbiome just can't function, right? So there are microbes in the seed that when that plant imbibes, when it takes in water and starts to germinate, those microbes come to life and start interacting with the soil around them to turn that into a living thing as well. And what do? We put poisons on the seed. So think about it. The other thing, I'm going to come back to the core microbiome in a minute. The other thing is biological induction. So that's where we have a plant, and maybe even one that we nuked with all kinds of horrible things on the seed, and it somehow survived and somehow germinated and grew, and then we had to keep feeding it to get it to grow. And then sometimes maybe it gets a chance to actually talk to the microbes that are in the soil. And some free-living microbes in the soil can be invited by the plant to come and form a relationship with them. And they will move from the soil into the plant and they'll spend the rest of their lives in that plant. Or they may leave again. So there's this constant moving. It is a holobiome. So biological induction is where something that was free-living in the soil has been invited by the plant to come and live in the plant. And most commonly, that's probably going to be a nitrogen-fixing organism. So we know that all green plants are capable of fixing nitrogen. It doesn't have to be a legume. In fact, most recent research shows you're probably better off in your mixes not having legumes in there. So if we look at the things that work, we can see out the window there, like some trees and stuff, probably no one's fertilising anything out there. So everything that's green that's out there has chlorophyll in its leaves, which is a part of a protein complex. That's the green pigment that absorbs light. And it cannot be green unless it has formed some kind of a relationship with nitrogen-fixing bacteria. And probably a lot of those are living in the plant and have been invited to come into the plant from the soil. So we see examples of, I may or may not talk about the Haggertys this afternoon, depends how much time I've got, but they're growing wheat on 40,000 hectares now with no nitrogen at all. They're out yielding all their neighbours. They've got much better high quality than their neighbours. They're producing nitrate-free grain, which they can sell into the Asian market for a premium price. And nitrate-free is going to be the way that things seem to be going in the commodity market. I thought it would have been glyphosate-free or chemical residue-free or something, but nitrate-free seems to be the way things are going, because nitrate is a potent carcinogen for humans. We don't want any nitrate in our food, and unfortunately we get a lot of nitrate in our food. Now I've got right off the track. So you can do a soil test on the Haggertys, and the soil test will show there's virtually no nitrogen in there, and agronomists will tell you there's no way you could grow a viable wheat crop here, and yet they're out yielding their neighbours. But when we look at the tissue tests, at the leaf tests, we find they're right in the sweet spot for nitrogen. They've got exactly the right amount of nitrogen in the leaves and good amounts of nitrogen, high protein in the grain. So what's happening there is biological induction. Those wheat plants are actually taking up nitrogen-fixing microbes from the soil, so you're not going to find it in the soil, you're only going to find it in the plants. So let's just go back to the core microbiome. This is a big topic we could actually talk about all day, so I'm just going to try and keep it as simple as I can. I'm not trying to dumb it down, but every single species of plant and every single cultivar within a species, in other words, every variety, has a specific assembly of bacteria, archaea, protists and fungi. They're all the different kinds of little things that we can't see with the naked eye. A specific grouping of those that actually belongs to that variety. So if you wanted to get diversity, for example, which I'm going to talk about later, one of the things you can even do is plant a whole lot of varieties of things. So if you were just going to grow wheat and you... I'm not so sure about whether you could do this with sugarcane, you probably can't, but if you were going to grow, let me think of some tropical things then, sunflower or sorghum or cowpeas or something, mung beans, or, sorry, I have to get my mind around tropical plants now. But if you were just going to grow one monocrop, a monocrop of something, one way you can get diversity simply, some diversity anyway, is just to put a whole lot of varieties in together of that crop, because every one of those will have its own core microbiome, and that will introduce a little bit of diversity. So those microbes are in the seeds, and then they form a relationship with the newly germinating plants, and then they move from the roots around that plant back into the stems and the leaves, and if it has flowers, and eventually the seeds of from where they're going to begin the cycle all over again. And in native natural communities, like native grasslands or prairies or something like that, or European alpine meadows, things like birds and little burrowing animals and stuff will actually move those seeds around sometimes, and when they do, they take that core microbiome as well. So whenever they distribute those seeds, then those core microbiome, and then wherever that seed ends up, it's going to start its new life with its own microbiome. So we have our own microbiomes too. And I mean, as we know now in human health, if there's some kind of intervention at birth and you don't get the microbiome from your mum, then it can create issues for the baby. So it's really important that babies do get the microbiome from their mothers. And it's very important that plants pass on their microbiomes to their seeds. And that microbial assembly remains within the plants throughout their life, and it's very important because it stimulates germination. Plants that have a good core microbiome will germinate faster and be more healthy. They'll have better root systems. They will actually get lots more nutrients from the soil. So again, we really interrupt that process when we put synthetic nutrients around those seeds. And it will increase its tolerance to biotic and abiotic stresses. So biotic stresses are things like, insect attack or fungal attack or something like that. And abiotic is like, a frost or a drought or waterlogging or anything that happens that's like a physical thing that happens to that plant. So the core microbiome can actually help the plant deal with all those kinds of stresses. So biological induction, this is the one where we have the movement of free-living microbes from the soil into the plants, and then they can stay for the rest of the plant's life if they decide that's what they want to do. And they're very significant, as I explained, for nitrogen fixing, for plant protection and for plant fitness. So what we've tried to do in the last 50 years or so, we've actually tried to replace all that biological activity with chemical fertilisers. We think, well, you know, plants can't grow without nitrogen, we know that, so why don't we just go and buy it and put it out there and, you know, we'll help them to grow. We know that plants can't grow without phosphorus, so we'll just go and add it and help them to grow. But we've actually got in the way, unfortunately, without knowing it. I mean, it wasn't deliberate. It's not a conspiracy theory. It's just ignorance on the part of humans. Because those plants that are supported by those high analysis fertilisers, and yes, they will grow, but they can't get the minerals and trace elements that they need to defend themselves. You'll have all kinds of other input costs then, like insecticides and fungicides, and then having to, you know, sometimes supply trace elements. They'll fall over in a windstorm or when it gets waterlogged, or if it's dry, you know, they can't grow as well. So you've really got a plant that's not fit. Its fitness has been reduced. So we have to think about now, with what we know about microbes, is actually supporting microbes to do all those kinds of jobs for us rather than using high analysis fertiliser. So the options that we have for that are to increase plant diversity, because that really stimulates that. We want to form that superorganism in the soil. We want to get that really humming with lots of different kinds of communities of microbes, lots of diversity in there, lots of conversations going on and communication. We can help that a little bit with a biostimulant. My thinking is that diversity is more important, but a biostimulant is, if you can't move to biodiversity easily or straight away, then that's a kind of transitional step. And the very best results that we're seeing are a plant diversity and a biostimulant together. So in my other presentations this afternoon, I'm going to talk about the extraordinary power of diversity, and then this afternoon I'm going to talk a little bit about biostimulants and various approaches to that. So we have a couple of minutes, I think we have 10 minutes left now for questions. There will also be a Q&A session after my diversity talk today and after the biostimulant talk. So there'll be an opportunity, if you have a question about diversity or about biostimulant, maybe wait for those sessions to ask those questions. So if you have something about microbes that you'd like to talk about now, we've got 10 minutes to do that. Oh, and Julia's did you explain about how this works?
I haven't had a chance yet, but we've got a new toy to play with to hopefully encourage you all to share your ideas and share comments or questions with us today. So this is a throwable microphone. We just pass it along, and if you feel like you want to say something, you say something, and you can direct your questions straight to Christine. So I throw that now right at you, so be ready. Somebody catch it, please. Oh, save the book.
That's all right.
If you want to say something, otherwise just pass it on.
I actually did have a question, but it's all wet now. No, that's all right. What's the ratio of plant root mass to rhizal mass? Is it that 99%?
So the question was, what's the ratio of plant root mass to rhizosphere mass? Sorry, this is feeding back for some reason.
That's probably water. It's all right. Yeah, so when you had that
so when I was talking about the census of life on Earth, and I said that most of the weight was in plants, you mean, and only...
No, with the little pine seedling where you had sort of equal amount of mass above the and then the rhizosphere below. What's the ratio of that root?
Oh, okay, so the question was, in the pine seedling where we had the little pine seedling and the roots were roughly equal to the tops, and then you've got all the, well, not only the mycelium that you can see, but all the bacterial colonies associated with that. They don't really weigh very much in that situation. I mean, the plant is going to weigh far more in that particular situation. If you weighed all those fungal hyphae, and they wouldn't it wouldn't wave it, because we can't even see them. Well, we can see those ones on pine trees, but the ones that are under all of our crops and under our pastures, we can't. They're there, but we can't see them.
I guess even in a three-dimensional sense, how much larger would that be? It seemed like huge, like compared to the root mass, because you've just got this.
In terms of the volume of it, it is huge.
Yeah, probably better
And
terminology.
you know, like, sorry, we might going to have to move that thing away because it's just feeding back.
Yeah, that's cool.
It's going really positive, yeah. I think
Okay, right, so that's better. Yes, in terms of the weight of the microbes associated with it, it'd be like with us. I mean, in terms of the weight of the microbes associated with our body, even though they outnumber us and we are 10% human on a cell count, but we're, 99.9% human by weight. So it would be the same. They don't weigh very much around that particular plant. The weight of them is not very much. Yes, yeah, and the numbers of them, like trillions, literally trillions of them. Trillions per cubic centimetre even. Like just 1 cubic centimetre of soil around a really healthy plant root, 1 centimetre by 1 centimetre, is going to have not billions but trillions. Remember, there's only 7 billion humans on the planet and there's trillions of things just living in 1 cubic centimetre of soil just right near the root plant. So there's lots of them by number, if that answers the question.
Yeah, Christine. We've listened to your podcast and we've changed our farming system so we're off synthetic fertilisers now. And we've noticed we're getting a lot of native species coming up into the cane. Would that be classed as our plant diversity coming into the system?
What do you mean by native species?
We're getting joint venture like legumes, all beneficials. They're not destroying the cane crop. They're not competing against the cane.
Are these native plants you're talking about, or are these invasive plants like weeds?
No,
If I can...
good succession of weeds or plants. They're beneficial.
Can I just pass it over to you, Simon? Because I'm not really sure I'm not familiar with those plants, I'm sorry.
watch out, the microphone is coming. Oh, not to me.
Watch out for the water.
Yeah, I guess there is there's a lot of native legumes in this country. Joint veg, I think you'll find it actually isn't a native as such, it's a naturalised legume, and there's quite a few of those as well. What's that other little red flowering one, Michael, that we see quite a lot of in our cane? What's it called? It's on the tip of my tongue. Nat?
Bethlehem?
No, that's a... Phasey bean, that's the one I was trying to think of. So Michael and John are seeing a lot more of this joint veg and phasey bean and those kinds of plants since they stopped using synthetic fertilisers.
Okay, so they're both legumes.
Yes.
They are pretty much, I'm pretty sure they're introduced. I don't think those two are
naturalised.
Yeah, they're naturalised. Mm. So if you're using nitrogen fertiliser, it will be very inhibitory to legumes. Like if you want to get rid of legumes out of a dairy pasture, for example, if you put nitrogen fertiliser on it and you've just planted red clover and white clover and all kinds of things, that you'll just lose them. So all the research shows that if you, in a pasture, I know it's a different situation, but if you have a grass legume pasture and you add nitrogen fertiliser, you lose the legumes. So the fact that you've stopped using the nitrogen fertiliser means the legumes have a chance to come back, have an opportunity to come back. So obviously the seeds were there, so you're now giving them an opportunity to come back. That's the only insight that I can provide to that question.
We're building a natural nitrogen fixing into the system.
Yes.
If we're not using nitrogen, everything's healthy, so something must be working.
Oh, I see what you're... So you're saying is the nitrogen coming from those? My take on that was that you're not inhibiting those plants anymore, because adding nitrogen fertiliser will definitely inhibit legumes. There's no doubt about that.
We weaned off the nitrogen, off the urea, but the system is still reducing.
Right. But you actually don't need... I see the question now. You don't need legumes in the system to produce nitrogen, because cane is perfectly capable of producing its own.
Yeah, well, that's what we're trying to figure.
I mean, like cane, so what I'm talking about with endophytes, there are a huge number of nitrogen-fixing endophytes living inside cane. It's known, like it's known throughout the world for having probably the highest concentration of nitrogen-fixing endophytes of any plant. And again, like what happens if you apply nitrogen fertiliser to a legume is that it stops it from fixing nitrogen. Right? So if you apply nitrogen fertiliser to cane, you stop it from fixing its own nitrogen. It can fix its own.
So what we're doing is working then?
What you're doing is working, but what you're seeing when you see the legumes coming in, they're responding to the fact that you haven't got high levels of available nitrogen in the soil. You don't want it in the soil. You never want to have high levels of available N in the soil. Like a soil test is completely upside down. Soil test should read 0 N, 0 P, and then a leaf test should show optimum N, optimum P, which show that everything is working inside the plant. Rather, it's totally inefficient to have it happening in the soil and then trying to get it from the soil into the plant. You know, we apply a highly mobile nutrient like nitrogen or something like phosphorus that gets fixed within hours sometimes of being, so it's highly immobile, but it gets fixed within the soil, becomes unavailable because it's a very highly reactive element, and it just combines with something else like iron or aluminium or calcium or something, gets taken out of circulation. So you spend all that money putting it on, and then it just gets, you know, absorbed within the soil matrix, and that's the end of it. Or we put a highly mobile nutrient on like nitrogen that the plant gets about 10% of, and the rest of it ends up in the air or out on the reef. And all we've done is interfere with the communication in the soil. So what you're doing is great, that you've got off urea and you're seeing things coming back to a more natural kind of communication system happening there. But I would suggest that the cane is fixing its own nitrogen, and the legumes you're seeing is just because you've stopped inhibiting them by using nitrogen fertiliser. Yes, there'll be beneficial interactions go on between those legumes and the cane, but that's probably not the main thing that's happening there.
Simon, do you want to pass it on? Or maybe
Can I just
David? Yep.
just a question, Christine, you mentioned before that legumes might be detrimental in some situations. Could you elaborate on that, please?
That surprised me as well when I saw the research on that. So that comes out of some German research, which I'm going to speak about in my next session. And that's a 15-year biodiversity experiment that's been running in Germany. And what they found was that when they compared a whole lot of combinations of plants, up to 60 different kinds of plant species in mixes, that they were looking at the rate of soil carbon accumulation in those soils, because that linked to everything else, as we know. So more carbon meant more available nitrogen, more available phosphorus, more available other nutrients, more life in the soil, better soil structure, better water holding capacity, all of those things. And then they looked at how quickly those mixes were putting carbon into the soil, and they found that the mixes that didn't have legumes in them, they were diverse mixes, but they didn't have legumes in them, built soil carbon much faster than the other mixes, which they thought was strange, because if we've got in a low diversity situation, like if you just have a grass like cane, for example, or any other kind of a grass like sorghum or maize or something, and you put a legume with it, you will usually see an improvement. Like if you plant soybeans down the same row as corn or something, for example, you'll usually see an improvement in the corn. Or faba beans. I don't know whether it's too hot for faba beans here, but they're fantastic with corn. Put them together. So we've always sort of seen that improvement by putting a legume in there, but what they found was with their really diverse mixes, if they had legumes in there, they didn't perform as well. So a grass and a legume is always going to be better than just a grass. But if you've got a lot of different species growing in together, you're better off not having the legumes in there. Because what happened was that the legumes had lots of bacteria around their roots. So they form bacterially dominated soil, whereas the other mixes that didn't have legumes in them had fungally dominated soils, and the fungally dominated soils were able to stabilise the carbon. And so you've got long-term carbon storage happening faster. It happened faster and it happened at a higher rate when there weren't legumes in there. So if you just have a grass-based system, you're going to be better off with some legumes in there. I'm not saying legumes are bad, but if you're looking at a really diverse mix, I'm encouraging people now to look at as many non-grass things as they can in their mixes and leave the legumes out. Or just have a few. Don't concentrate lots. I mean, I could just like to give you an example from vegetable farmers. Organic growers in California, for example, have been, for years, they've just had monospecific covers of something like vetch. And it'll be beautiful. It'll be this high and there'll be tons of it, like, 10 tonnes, like irrigated, 20 tonnes of vetch. And then they turn it all back in and then they'll plant tomatoes or something after that. And then when I talk about the liquid carbon pathway and, green plants and photosynthesis and everything, I say, Christine, you don't know what you're talking about. We've been growing covers for 20 years and our soil carbon hasn't increased. I say, what were your covers? Well, they're monocrops of legumes. Well, that's why your soil carbon hasn't increased. And soybean has a reputation on its own as being one of the most degrading things that you can plant in soil. If you plant soybean crops 3 years in a row, like in the United States in the corn soy belt, 3 years in a row of soybeans, you've almost totally destroyed your soil. So think about that. You do not ever want a monoculture of legumes, and you certainly, if you're looking at diverse mixes, just keep the legumes down. Sometimes they're handy to have in there, but just don't. We've just thought legumes and N, we've related legumes and nitrogen. We've forgotten about the endosphere, for example, the fact that plants can take free-living nitrogen fixes from the soil and cultivate them within their own systems, or things like sugarcane is well known as for having lots of endophytes, nitrogen-fixing endophytes within the system itself.
Christine, could I just add a little something to that? Your nitrogen, your legumes, by and large, particularly your annual legumes, will be the fibre within that plant is high carbon nitrogen ratio. So,
high nitrogen to carbon, yes. Or low carbon to nitrogen.
Yes. Yeah. So typically soybeans, that biomass will be 30 to 1, whereas sugarcane will be 85 or 90 to 1. So I would suggest that that's one of the reasons why your legumes aren't actually transferring carbon, as much carbon, to the soil, because they don't consist of it in the first place.
Yeah, and they break down really quickly as well.
Yes.
Like legumes.
Well, that's why they disappear.
Yeah, like
because they have no
exactly.
fibre. So
someone growing a huge veg crop and it's all lush and green, they turn it in a very short amount of time, especially in an irrigated system. And if it's warm and moist, well, I mean, it's just going to be gone. Legume residues don't stay around.
No.
They're very rapidly decomposed. So nothing really from them will remain in the soil. So, it's a useful thing to have in a mix, but unfortunately we've placed a lot of emphasis on legumes because we haven't actually understood how these microbial systems work within plants. So all I'm saying is, I'm not saying legumes are bad, I'm saying just don't put all your emphasis on legumes, and remember, if you've got diversity, you'll have plenty of N. And I'll talk more about diversity, and these questions probably really more fit into when we talk about diversity later.
I'm not going to throw it that far.
Christine, with the tropical grasses, has there been any work done on combinations of grasses that perform better in building soil than others?
So what we've seen traditionally with our tropical grasses as well is for combining them with, stylo or something, like the thing has been to put legumes with them. And obviously that is going to be better than just the grasses on their own. I mean, we can see that a little bit of an improvement. I mean, the thing about diversity, and really these questions belong in the diversity session, because we've got lots of things we could talk about, what you could put together and whatever with plant diversity. But we want to start looking at different functional groups of plants, which I am going to cover in my next talk. That as many diversities of, as much variety of functional group, really different kinds of plants from different plant families, I mean. So the thing with grasses is, yes, you could put different kinds of tropical grasses together. You could even put different varieties of the one thing together, and it is going to be a slight improvement to just having one variety of it, definitely. I mean, even with things like green panic, for example. You know, there's green panic and Gatton panic, and like there's several varieties of panic, and I know that the guys in Western Australia that have gone into putting perennial grasses into what was an annual system have found if they put different varieties of panic together, they will get a much better result, because they grow slightly different times of the year, etc. So there's some advantage to that, but there is a huge advantage to having more functional groups than that. So really probably need to start looking at things that outside the grass family and outside the legume family even, for what could go into those mixes. We have been very tunnel vision in our view of what to plant. So is there any questions about microbes? Oh. No? Yes?
So I think your answer hopefully will be related to microbes. My question is about Pasture dieback and what your thoughts are related to the microbe kind of involvement with that.
Okay, so I get a question about Pasture dieback every time I come to Queensland, and I don't know anything about it, but I'm not the only one that doesn't know anything about it, I gather. No one knows what's going on. So my personal opinion on that, just my opinion, is that you've just got, like, it's mostly the buffel, I gather. Is that right?
It's now in pretty much all the natives and all improved pastures.
All the
natives, as well as everything else. We haven't really seen it hit anything, but, well, yeah, it's just in everything.
So it's, well, okay, I thought it was more, like, so it originally started in the buffel, didn't it? And I thought, that's just because you've got a monoculture and there's not enough diversity there. That was what I was thinking. But there hasn't been any organism, like, what's the word for it? They haven't been able to isolate anything, any organism. So it'd be interesting to see what would happen if, like, say, an area where everything had died back, if a really diverse mix was put in there, like 20 species. You have to remember that the original grasslands and grassy woodlands that were here were, apart from the areas that were tropical rainforest, of course, but the other areas that were savannas, there was like 500 to 700 different ground cover plants in those, which is what I'm going to be talking about in my next session. So you go from 500 to 700 plants to just a domination of something like buffel, or even, most of the things you could just count on one hand what's there. So I really think it's lack of diversity, so we'll talk more about that. So this lady here actually had a question. I know, well, it probably wasn't about microbes, but
no,
Julia's
about plant diversity, and I thought, well, we're better off just having, if you've got grass and the weeds, well, the weeds are part of the diversity, yes?
Well, the weeds are nature's solution to the fact that you just had grass. Like, nature abhors a vacuum, and just grass is it's not as bad as bare soil. I mean, it's definitely better, but like I said, in my next talk, I'll talk about the fact that there were hundreds and hundreds, literally hundreds, of different things all growing together in plant communities, and all the communication that was going on there. Of course, when we simplify that, we're going to run into problems. And I just see the pasture dieback as being symptomatic, really, of lack of microbial diversity. In the end, the whole system is going to fall over if we're not supporting those microbes, because microbes run the place. And if we look at, again, I'm talking about preempting what I'm going to talk about later, but, the human system, we know that the quality of our food and the diversity of our food is really important for our microbiome. We know that for ruminants, the quality of food and the diversity of what they're consuming is incredibly important for their rumen function. And for soil, it's the quality of food and the diversity of food that is really important for the soil microbiome to function. And what is food for soil microbes? It's plant root exudates. And if you don't have lots of different kinds of plant root exudates supporting lots of different functional groups, the soil can't function. And when the soil can't function, everything above it is going to fall over. And I think that's what we're seeing. Because if there's been nothing being able to be isolated, it's not a pathogen that's causing the problem. Soil has basically just fallen over and died. And then the plants can't grow. So that would be my opinion, with nothing to back it up. But so the end of this session.
Yes, thank you, Christine, for your presentation.
So I'm going to be taking a break during this break, because I'm not the next speaker, am I? But then I do have another talk after that.
That's right.
So if you could please just keep your questions to Q&A times, because we have another Q&A session after my next talk, and then another one later this afternoon. And then if there's still more questions at the end of the day, I'm happy to answer them now. But if I could have a little break now, that would be lovely. Thank you.